Banana is an elongated, edible fruit, botanically a berry, produced by several kinds of large tree-like herbaceous flowering plants in the genus Musa. In some countries, cooking bananas are called plantains, distinguishing them from dessert bananas. The fruit is variable in size, color, and firmness, but is usually elongated and curved, with soft flesh rich in starch covered with a peel, which may have a variety of colors when ripe. It grows upward in clusters near the top of the plant. Almost all modern edible seedless parthenocarp cultivated bananas come from two wild species, Musa acuminata and Musa balbisiana, or their hybrids. Musa species are native to tropical Indomalaya and Australia. They were probably domesticated in New Guinea. They are grown in 135 countries, primarily for their fruit, and to a lesser extent to make banana paper and textiles, while some are grown as ornamental plants. The world's largest producers of bananas in 2022 were India and China, which together accounted for approximately 26% of total production. Bananas are eaten raw or cooked in recipes varying from curries to banana chips, fritters, fruit preserves, or simply baked or steamed. Worldwide, there is no sharp distinction between dessert bananas and cooking plantains. This distinction works well enough in the Americas and Europe, but it breaks down in Southeast Asia, where many more kinds of bananas are grown and eaten. The term banana is applied also to other members of the Musa genus, such as the scarlet banana, Musa coccinea, the pink banana, Musa velutina, and the fay bananas. Members of the genus Enceat, such as the snow banana, Enceat glaucum, and the economically important false banana, Enceat ventricosum of Africa, are sometimes included. Both genera are in the banana family, Musaceae. Banana plantations can be damaged by parasitic nematodes and insect pests, and to fungal and bacterial diseases, one of the most serious being Panama disease, which is caused by a fusarium fungus. This and black sigatoka threaten the production of Cavendish bananas, the main kind eaten in the Western world, which is a triploid Musa acuminata. Plant breeders are seeking new varieties, but these are difficult to breed given that commercial varieties are seedless. To enable future breeding, banana germplasm is conserved in multiple gene banks around the world. Description: acorn, about 25 centimeters, 10 in, across shutterstock. Young plant female flowers have petals at the tip of the ovara tree. Showing fruit and inflorescence. Single row planting inflorescence. Partially open. The banana plant is the largest herbaceous flowering plant. All the above ground parts of a banana plant grow from a structure called a corm. Plants are normally tall and fairly sturdy with a tree-like appearance, but what appears to be a trunk is actually a pseudostem composed of multiple leaf stalks, petioles. Bananas grow in a wide variety of soils, as long as it is at least 60 centimeters, 2 ft deep, has good drainage, and is not compacted. They are fast-growing plants, with a growth rate of up to 1.6 meters, 5.2 ft per day. The leaves of banana plants are composed of a stalk, petiole, and a blade, lamina. The base of the petiole widens to form a sheath. The tightly packed sheaths make up the pseudostem, which is all that supports the plant. The edges of the sheath meet when it is first produced, making it tubular. As new growth occurs in the center of the pseudostem, the edges are forced apart. Cultivated banana plants vary in height depending on the variety and growing conditions. Most are around 5 m, 16 ft tall, with a range from dwarf Cavendish plants at around 3 m, 10 ft, to Gros Mitchell at 7 m, 23 ft or more. Leaves are spirally arranged and may grow 2.7 meters, 8.9 ft, long and 60 centimeters, 2 ft wide. When a banana plant is mature, the corm stops producing new leaves and begins to form a flower spike or inflorescence. A stem develops which grows up inside the pseudostem, carrying the immature inflorescence until eventually it emerges at the top. Each pseudostem normally produces a single inflorescence, also known as the banana heart. After fruiting, the pseudostem dies, but offshoots will normally have developed from the base, so that the plant as a whole is perennial. The inflorescence contains many petal-like bracts between rows of flowers. The female flowers, which can develop into fruit, appear in rows further up the stem, closer to the leaves from the rows of male flowers. The ovary is inferior, meaning that the tiny petals and other flower parts appear at the tip of the ovary. The banana fruits develop from the banana heart, in a large hanging cluster called a bunch, made up of around 9 tiers called hands, with up to 20 fruits to a hand. A bunch can weigh 22 to 65 kilograms, 49 to 143 pounds. The stalk ends of the fruits connect up to the rachis part of the inflorescence. Opposite the stalk end is the blossom end, where the remnants of the flower deviate the texture from the rest of the flesh inside the peel. The fruit has been described as a leathery berry. There is a protective outer layer, a peel or skin, with numerous long, thin strings, vascular bundles, which run lengthwise between the skin and the edible inner white flesh. The peel is less palatable and usually discarded after peeling the fruit. The inner part of the common yellow dessert variety can be split lengthwise into 3 sections that correspond to the inner portions of the 3 carpels by manually deforming the unopened fruit. In cultivated varieties, fertile seeds are usually absent. Evolution Phylogeny 2011 phylogenomic analysis using nuclear genes indicates the phylogeny of some representatives of the Musaceae family. Major edible kinds of banana are shown in boldface. Musaceae Ensete ventricosum, Ensete or false banana of Africa Musella laciocarpa, golden lotus banana of China Musiclade, and Musa balbisiana, plantain of South, East, and Southeast Asia Musa mannii, a wild banana of Arunachal Pradesh, India Musa acuminata ssp. Zebrina, blood banana of Sumatra Musa acuminata ssp. Burmanica, banana, S. India to Cambodia Musa ornata, flowering banana of Southeast Asia Clade A. Musa coccinea, scarlet banana of China and Vietnam Musa becquerel, a wild banana of Sabah Musa textilis, abaca or Manila, hemp of the Philippines Musa macleayi of Papua New Guinea and Solomon Islands Musa X troglodytarum. Fay banana of French Polynesia, many cultivated bananas are hybrids of M. acuminata x M. balbisiana, not shown in tree. Work by Lee and colleagues in 2024 identifies 3 subspecies of M. acuminata, namely SSPP, Banksii, Malaxensis, and Zebrina, as contributing substantially to the BAN, DH, and Z subgenomes of triploid cultivated bananas, respectively. Taxonomy Musa nendron, cultivar, grown widely in the Indian state of Kerala. Further information. List of banana cultivars. The genus Musa was created by Carl Linnaeus in 1753. The name may be derived from Antonius Musa, physician to the Emperor Augustus, or Linnaeus may have adapted the Arabic word for banana, maz. The ultimate origin of Musa may be in the Trans, New Guinea languages, which have words similar to muku. From there, the name was borrowed into the Austronesian languages and across Asia, accompanying the cultivation of the banana as it was brought to new areas, via the Dravidian languages of India, into Arabic as a wanderwort. The word banana is thought to be of West African origin, possibly from the Wolof word banana, and passed into English via Spanish or Portuguese. Musa is the type genus in the family Musaceae. The APG-III system assigns Musaceae to the order Zingiberales, part of the camelonid clade of the monocotyledonous flowering plants. Some 85 species of Musa were recognized by Plants of the World Online as of July 2025. Several produce edible fruit, while others are cultivated as ornamentals. The classification of cultivated bananas has long been a problematic issue for taxonomists. Linnaeus originally placed bananas into 2 species based only on their uses as food: Musa sapientum for dessert bananas and Musa paradisiaca for plantains. More species names were added, but this approach proved to be inadequate for the number of cultivars in the primary center of diversity of the genus, Southeast Asia. Many of these cultivars were given names that were later discovered to be synonyms. In a series of papers published from 1947 onward, Ernest Cheeseman showed that Linnaeus's Musa sapientum and Musa paradisiaca were cultivars and descendants of 2 wild seed-producing species, Musa acuminata and Musa balbisiana, both first described by Luigi Aloysius Calla. Cheeseman recommended the abolition of Linnaeus's species in favor of reclassifying bananas according to 3 morphologically distinct groups of cultivars, those primarily exhibiting the botanical characteristics of Musa balbisiana, those primarily exhibiting the botanical characteristics of Musa acuminata, and those with characteristics of both. Researchers Norman Simmons and Ken Shepard proposed a genome-based nomenclature system in 1955. This system eliminated almost all the difficulties and inconsistencies of the earlier classification of bananas based on assigning scientific names to cultivated varieties. Despite this, the original names are still recognized by some authorities, leading to confusion. The accepted scientific names for most groups of cultivated bananas are Musa acuminata Kala and Musa balbisiana Kala for the ancestral species, and Musa times paradisiaca L for the hybrid of the two. An unusual feature of the genetics of the banana is that chloroplast DNA is inherited maternally, while mitochondrial DNA is inherited paternally. This facilitates taxonomic study of species and subspecies relationships. In formal classification in regions such as North America and Europe, Musa fruits offered for sale can be divided into small sweet bananas, eaten raw when ripe as a dessert, and large starchy, plantains, or cooking bananas, which do not have to be ripe. Linnaeus made this distinction when naming two species of Musa. Members of the plantain subgroup of banana cultivars, most important as food in West Africa and Latin America, correspond to this description, having long pointed fruit. They are described by Ploetz et al. As true plantains, distinct from other cooking bananas. The cooking bananas of East Africa belong to a different group, the East African highland bananas. Further, small farmers in Colombia grow a much wider range of cultivars than large commercial plantations do, and in Southeast Asia, the center of diversity for bananas, both wild and cultivated. The distinction between bananas and plantains does not work. Many bananas are used both raw and cooked. There are starchy cooking bananas, which are smaller than those eaten raw. The range of colors, sizes, and shapes is far wider than in those grown or sold in Africa, Europe, or the Americas. Southeast Asian languages do not make the distinction between bananas and plantains that is made in English. Thus, both Cavendish dessert bananas and Sabah cooking bananas are called pisang in Malaysia and Indonesia, Kluai in Thailand, and Chu letter 1ed1I in Vietnam. Fei bananas, grown and eaten in the islands of the Pacific, are derived from a different wild species. Most Fei bananas are cooked, but carrot bananas, which are short and squat with bright red skins, are eaten raw. History original native ranges of the ancestors of modern edible bananas. Red, Musa acuminata. Blue, Musa balbisiana domestication. See also Musa acuminata, domesticated plants and animals of Australasia, and East African highland bananas. Fruits of wild-type bananas have numerous large, hard seeds. The earliest domestication of bananas, Musa spp, was from naturally occurring parthenocarpic, seedless, individuals of Musa banksii in New Guinea. These were cultivated by Papuans before the arrival of Austronesian speakers. Numerous phytoliths of bananas have been recovered from the Cuck Swamp archaeological site and dated to around 10,000 to 6,500 BP. Foraging humans in this area began domestication in the late Pleistocene using transplantation and early cultivation methods. Various investigations, including Denham et al. 2003, determined that by the early to middle of the Holocene, the process was complete. Ancient spread Austronesian trade routes from New Guinea. Cultivated bananas spread westward into island Southeast Asia. They hybridized with other, possibly independently domesticated, subspecies of Musa acuminata, as well as M. balbisiana in the Philippines, northern New Guinea, and possibly Halmahera. These hybridization events produced the triploid cultivars of bananas commonly grown today. From island Southeast Asia, they became part of the staple domesticated crops of the Austronesian peoples, and were spread via their ancient seaborne migrations and ancient maritime trading routes into Oceania, Africa, South Asia, and Indochina. Chronological dispersal of Austronesian peoples across the Indo-Pacific bananas are believed to have been introduced to Africa from Southeast Asia via the Austronesian settlement of Madagascar, c. 600 AD. This is supported by relict populations of accidentally introduced, seeded Musa acuminata in northeastern Madagascar and Pemba Island, off Tanzania, as well as the phenotypes of cultivated polyploid bananas and their distribution, all of which provide the clearest evidence of a Southeast Asian origin. However, the role of Madagascar as a staging board for the dispersal of bananas and other Asian crops is unclear. Malagasy people colonized Madagascar from island Southeast Asia around 600 AD onwards, but contact between East Africa and island Southeast Asia dates back to at least 300 BC or earlier. The possibility that bananas may have been introduced from earlier Austronesian settlements in the East African coast that predate the settlement of Madagascar cannot be ruled out. These ancient introductions resulted in the banana subgroup, now known as the true plantains, which include the East African highland bananas and the Pacific plantains, the Iholna and Mauli-Papulu subgroups. Genetic evidence show that East African highland bananas, AAA, originated from banana populations introduced to Africa from the region between Java, Borneo, and New Guinea. Pacific plantains, AAB, on the other hand, were introduced to the Pacific Islands from banana populations originating from either eastern New Guinea or the Bismarck Archipelago. Another wave of introductions later spread domesticated polyploid bananas to other parts of tropical Asia, particularly Indochina and the Indian subcontinent. Southeast Asia remains the region of primary diversity of the banana. Areas of secondary diversity are found in Africa, indicating a long history of banana cultivation there. Other hypotheses, 21st century discoveries of alleged banana phytoliths in Uganda and Cameroon, dating to the first millennium BC, and earlier triggered a debate about the date of the first introduction of bananas to East Africa. However, the identification of the remains in Uganda as phytoliths, much less banana phytoliths, is now considered dubious. The Cameroon phytoliths, on the other hand, are confirmed as Musa, despite early doubts that they may be from Ncete. However, the incongruous early date, all other archaeobotanical remains of bananas in Africa being from at earliest the first millennium AD, remains questionable due to the low number of phytoliths recovered, 25, the absence of additional phytoliths in more recent sediments, and the possibility that the apparent date was the result of stratigraphic mixing. An introduction date of 2000 to 1000 BC is also unlikely, as this was long before there were any evidence of agriculture in East Africa. Polyploid banana cultivars are sterile and do not spread without human cultivation. Similarly, phytoliths recovered from the Khat Digi archaeological site in Pakistan were interpreted as evidence that bananas were known to the Indus Valley Civilization. This may indicate very early dispersal of bananas by Austronesian traders by sea from as early as 2000 BCE. But this is still putative, as they may have come from local wild Musa species used for fiber or as ornamentals, not food, and banana phytoliths are absent in other contemporary sites in South Asia. Glucanase and two other proteins specific to Musaceae were found in dental calculus from the early Iron Age, 12th century BCE, Philistines in Tel Irani in the southern Levant. However, the authors only tentatively identify it as Musa, as the proteins can also be found in Ncete, cultivated for their edible corms and pseudostems in Africa. Arab Agricultural Revolution. Further information. Arab Agricultural Revolution. Actual and probable diffusion of bananas during the Arab Agricultural Revolution, 700 to 1500 C. The banana may have been present in isolated locations in the Middle East on the eve of Islam. The spread of Islam was followed by far-reaching diffusion. There are numerous references to it in Islamic texts, such as poems and hadiths, from the 9th century onwards. By the 10th century, the banana appeared in texts from Palestine and Egypt. From there, it diffused into North Africa and Al-Andalus, Islamic Spain during the Arab Agricultural Revolution. An article on banana tree cultivation is included in Ibn al-Awam's 12th century agricultural work, Kitab al-Fila Macron Letter 1E25, Book on Agriculture. During the Middle Ages, bananas from Granada were considered among the best in the Arab world. Bananas were certainly grown in the Christian kingdom of Cyprus by the late medieval period. Writing in 1458, the Italian traveler and writer Gabriel Capodilista wrote favorably of the extensive farm produce of the estates at Episcopi, near modern-day Limassol, including the area's banana plantations. Early modern spread. Further information. Columbian Exchange Illustration of Fruit and Plant, Acta Eruditorum, 1734. In the early modern period, Bananas were encountered by European explorers during the Magellan expedition in 1521, in both Guam and the Philippines. Lacking a name for the fruit, the ship's historian Antonio Pigafetta described them as figs more than 1 palm long. Bananas were introduced to South America by Portuguese sailors who brought them from West Africa in the 16th century. Southeast Asian banana cultivars, as well as abaca grown for fibers, were introduced to North and Central America by the Spanish from the Philippines via the Manila galleons. Plantation cultivation. Further information. History of modern banana plantations in the Americas. Plantation in the Philippines, 2010. In the 15th and 16th centuries, Portuguese colonists started banana plantations in the Atlantic Islands, Brazil, and Western Africa. North Americans began consuming bananas on a small scale at very high prices shortly after the Civil War, though it was only in the 1880s that the food became more widespread. As late as the Victorian era, bananas were not widely known in Europe, although they were available. The earliest modern plantations originated in Jamaica and the related Western Caribbean zone, including most of Central America. Plantation cultivation involved the combination of modern transportation networks of steamships and railroads with the development of refrigeration that allowed more time between harvesting and ripening. North American shippers like Lorenzo Dow Baker and Andrew Preston, the founders of the Boston Fruit Company, started this process in the 1870s with the participation of railroad builders like Minor C. Keith. Development led to the multinational giant corporations like Chiquita and Dole. These companies were monopolistic, vertically integrated, controlling growing, processing, shipping, and marketing, and usually used political manipulation to build enclave economies, internally self-sufficient, virtually tax-exempt, and export-oriented, contributing little to the host economy. Their political maneuvers, which gave rise to the term Banana Republic for states such as Honduras and Guatemala, included working with local elites and their rivalries to influence politics or playing the international interests of the United States, especially during the Cold War, to keep the political climate favorable to their interests. Small-scale cultivation. Further information. History of peasant banana production in the Americas. Small-scale banana production, Liberia, 2013. The vast majority of the world's bananas are cultivated for family consumption or for sale on local markets. They are grown in large quantities in India, while many other Asian and African countries host numerous small-scale banana growers who sell at least some of their crop. Peasants with small holdings of 1 to 2 acres in the Caribbean produce bananas for the world market, often alongside other crops. In many tropical countries, the main cultivars produce green, unripe bananas used for cooking. Because bananas and plantains produce fruit year-round, they provide a valuable food source during the hunger season between harvests of other crops, and are thus important for global food security. Modern cultivation. See also. List of banana cultivars. Bananas are propagated asexually from offshoots. The plant is allowed to produce 2 shoots at a time, a larger one for immediate fruiting and a smaller sucker or follower to produce fruit in 6 to 8 months. As a non-seasonal crop, bananas are available fresh year-round. They are grown in some 135 countries. Cavendish. Main article. Cavendish banana Shutterstock cultivars in the Cavendish Group dominate the world market. In Global Commerce in 2009, by far the most important cultivars belonged to the triploid Musa acuminata AAA group of Cavendish Group bananas. Disease is threatening the production of the Cavendish banana worldwide. It is unclear if any existing cultivar can replace Cavendish bananas, so various hybridization and genetic engineering programs are attempting to create a disease-resistant, mass-market banana. One such strain that has emerged is the Taiwanese Cavendish or Formisana. Ripening export bananas are picked green and ripened in special rooms upon arrival in the destination country. These rooms are airtight and filled with ethylene gas to induce ripening, which mimics the normal production of this gas as a ripening hormone. Ethylene stimulates the formation of amylase, an enzyme that breaks down starch into sugar, influencing the taste. Ethylene signals the production of pectinase, a different enzyme which breaks down the pectin between the cells of the banana, causing the banana to soften as it ripens. The vivid yellow color many consumers in temperate climates associate with bananas is caused by ripening around 18 degrees C, 64 degrees F, and does not occur in Cavendish bananas ripened in tropical temperatures over 27 degrees C, 81 degrees F, which leaves them green. Storage and transport Ralstonia solanacearum on an overripe bananas are transported over long distances from the tropics to world markets. To obtain maximum shelf life, harvest comes before the fruit is mature. The fruit requires careful handling, rapid transport to ports, cooling, and refrigerated shipping. The goal is to prevent the bananas from producing their natural ripening agent, ethylene. This technology allows storage and transport for 3 to 4 weeks at 13 degrees C, 55 degrees F. On arrival, bananas are held at about 17 C, 63 degrees F, and treated with a low concentration of ethylene. After a few days, the fruit begins to ripen and is distributed for final sale. Ripe bananas can be held for a few days at home. If bananas are too green, they can be put in a brown paper bag with an apple or tomato overnight to speed up the ripening process. Sustainability: the excessive use of fertilizers contributes greatly to eutrophication in streams and lakes, harming aquatic life, while expanding banana production has led to deforestation. As soil nutrients are depleted, more forest is cleared for plantations. This causes soil erosion and increases the frequency of flooding. Voluntary sustainability standards such as Rainforest Alliance and Fairtrade are being used to address some of these issues. Banana production certified in this way grew rapidly at the start of the 21st century to represent 36% of banana exports by 2016. However, such standards are applied mainly in countries which focus on the export market, such as Colombia, Costa Rica, Ecuador, and Guatemala. Worldwide, they cover only 8 to 10% of production. Breeding edible bananas are parthenocarpic, seedless. This is important for their edibility, but the lack of seed production makes breeding difficult. Furthermore, cultivated bananas are typically triploid, less commonly diploid, and a few tetraploid. They are typically derived from the wild diploid species M. acuminata and M. balbisiana, although some originate from M. acuminata only. Unpaired chromosomes in triploids, homeologous recombination between chromosomes from the two ancestral species, and failures to produce functional reproductive structures, due to selection for parthenocarpy, all combine to cause extremely low frequencies of successful seed production when attempting to breed bananas. Starting in the 1920s, pedigree breeding has been employed to try to generate desirable hybrids. Existing triploids are bred against diploids, sometimes wild with a desired characteristic, trying to create a tetraploid by combining a haploid gamete from the diploid parent with a rare, unreduced triploid gamete from the other. The resulting tetraploid can then be bred against another diploid to produce triploid offspring. More recently, the reproductive breeding strategy treats diploids with colchicine to produce tetraploid offspring, which can then be bred against a second diploid to produce triploids. Mutation breeding can be used in this crop. Aneuploidy is a source of significant variation in allotriploid varieties. For one example, it can be a source of TR4 resistance. Lab protocols have been devised to screen for such aberrations and for possible resulting disease resistances. Wild Musa SBP provide useful resistance genetics and are vital to breeding for TR4 resistance, as shown in introgressed resistance from wild relatives. The Honduran Foundation for Agricultural Research has bred a seedless banana that is resistant to both Panama disease and black sigatoka disease. The team made use of the fact that seedless varieties do rarely produce seeds. They obtained around 15 seeds from some 30,000 cultivated plants, pollinated by hand with pollen from wild Asian bananas. Production and export banana production tons, 2022 production, in millions of Republic of the Rica 2, 50, 12, 6 Ivory Coast 052 12 6 Mexico 262, 6 Dominican FAOSDAT of the United Nations. Note: some countries distinguish between bananas and plantains, but 4 of the top 6 producers do not, thus necessitating comparisons using the total for bananas and plantains combined. As of 2018, bananas are exported in larger volume and to a larger value than any other fruit. In 2022, world production of bananas and plantains combined was 179 million tons, led by India and China with a combined total of 26% of global production. Other major producers were Uganda, Indonesia, the Philippines, Nigeria, and Ecuador. As reported for 2013, total world exports were 20 million tons of bananas and 859,000 tons of plantains. Ecuador and the Philippines were the leading exporters with 5.4 and 3.3 million tons, respectively, and the Dominican Republic was the leading exporter of plantains with 210,350 tons. Pests. Bananas are damaged by a variety of pests, especially nematodes and insects. Nematodes. Banana roots are subject to damage from multiple species of parasitic nematodes. Radifolus similis causes nematode root rot, the most serious nematode disease of bananas in economic terms. Root knot is the result of infection by species of Melidogen, while root lesion is caused by species of Pratylenchus, and spiral nematode root damage is the result of infection by Helicotylenchus species. Radifolus similis inside banana root, causing nematode root rot. Insects. Among the main insect pests of banana cultivation are two beetles that cause substantial economic losses: the banana borer Cosmopolite sordidus and the banana stem weevil Odoporus longicalis. Other significant pests include aphids and scarring beetles. The banana borer is a destructive pest that tunnels inside the plant. Diseases. Main article. List of banana and plantain diseases. Although in no danger of outright extinction, bananas of the Cavendish group, which dominate the global market, are under threat. There is a need to enrich banana biodiversity by producing diverse new banana varieties, not just focusing on the Cavendish. Its predecessor, Gros Michel, discovered in the 1820s, was similarly dominant but had to be replaced after widespread infections of Panama disease. Monocropping of Cavendish similarly leaves it susceptible to disease and so threatens both commercial cultivation and small-scale subsistence farming. Within the data gathered from the genes of hundreds of bananas, the botanist Julie Sardos has found several wild banana ancestors currently unknown to scientists, whose genes could provide a means of defense against banana crop diseases. Some commentators have remarked that those variants which could replace what much of the world considers a typical banana are so different that most people would not consider them the same fruit, and blame the decline of the banana on monogenetic cultivation driven by short-term commercial motives. Overall, fungal diseases are disproportionately important to small island developing states. Panama disease, Fusarium fungus climbing up through the banana stem. Panama disease is caused by a Fusarium soil fungus, which enters the plants through the roots and travels with water into the trunk and leaves, producing gels and gums that cut off the flow of water and nutrients, causing the plant to wilt and exposing the rest of the plant to lethal amounts of sunlight. Prior to 1960, almost all commercial banana production centered on the Gros Michel cultivar, which was highly susceptible. Cavendish was chosen as the replacement for Gros Michel because, among resistant cultivars, it produces the highest quality fruit. It requires more care during shipping, and its quality compared to Gros Michel is debated. Fusarium wilt tr4, a reinvigorated strain of Panama disease, was discovered in 1993. This virulent form of Fusarium wilt has destroyed Cavendish plantations in several Southeast Asian countries and spread to Australia and India. As the soil-based fungi can easily be carried on boots, clothing, or tools, the wilt spread to the Americas despite years of preventive efforts. Without genetic diversity, Cavendish is highly susceptible to TR4, and the disease endangers its commercial production worldwide. The only known defense to TR4 is genetic resistance. This is conferred either by RGA2, a gene isolated from a TR4-resistant diploid banana, or by the nematode-derived SED9. This may be achieved by genetic modification. Black sigatoka leaf infected with black sigatoka is a fungal leaf spot disease first observed in Fiji in 1963 or 1964. It is caused by the Ascomycete Mycosphaerella fijiensis. The disease, also called black leaf streak, has spread to banana plantations throughout the tropics from infected banana leaves used as packing material. It affects all main cultivars of bananas and plantains, including the Cavendish cultivars, impeding photosynthesis by blackening parts of the leaves, eventually killing the entire leaf. Starved for energy, fruit production falls by 50% or more, and the bananas that do grow ripen prematurely, making them unsuitable for export. The fungus has shown ever-increasing resistance to treatment. Spraying with fungicides may be required as often as 50 times a year. Better strategies with integrated pest management are needed. Banana bunchy top virus colony of banana aphids, Pentalonia nigronervosa. Vector of banana bunchy top virus. Banana bunchy top virus is a plant virus of the genus Babuvirus, family Nanoviridae, affecting Musa spp. Including banana, abaca, plantain and ornamental bananas, and enceinte spp. In the family Musaceae. Banana bunchy top disease symptoms include dark green streaks of variable length in leaf veins, midribs, and petioles. Leaves become short and stunted as the disease progresses, becoming bunched at the apex of the plant. Infected plants may produce no fruit, or the fruit bunch may not emerge from the pseudostem. The virus is transmitted by the banana aphid Pentalonia nigronervosa and is widespread in Southeast Asia, the Philippines, Taiwan, Oceania, and parts of Africa. There is no cure, but it can be effectively controlled by the eradication of diseased plants and the use of virus-free planting material. No resistant cultivars have been found, but varietal differences in susceptibility have been reported. The commercially important Cavendish subgroup is severely affected. Banana bacterial wilt. Banana bacterial wilt is a bacterial disease caused by Xanthomonas campestris PV. Musacearum. First identified on a close relative of bananas, Enceinte ventricosum, in Ethiopia in the 1960s, the disease was first seen in Uganda in 2001, affecting all banana cultivars. Since then, it has been diagnosed in Central and East Africa, including the banana-growing regions of Rwanda, the Democratic Republic of the Congo, Tanzania, Kenya, Burundi, and Uganda. Conservation of genetic diversity. The cold storage room for the banana collection at Bioversity International's Musa germplasm transit center. Given the narrow range of genetic diversity present in bananas and the many threats via biotic pests and diseases and abiotic threats, such as drought, stress, conservation of the full spectrum of banana genetic resources is ongoing. In 2024, the economist Pascal Liu of the FAO described the impact of global warming as an enormous threat to the world's supply of bananas. Banana germplasm is conserved in many national and regional gene banks, and at the world's largest banana collection, the International Musa Germplasm Transit Center, managed by Bioversity International and hosted at KU Leuven in Belgium. Since Musa cultivars are mostly seedless, they are conserved by three main methods: in vivo, planted in field collections; in vitro, as plantlets in test tubes within a controlled environment; and by cryopreservation, meristems conserved in liquid nitrogen at -196 degrees C. Genes from wild banana species are conserved as DNA and as cryopreserved pollen. Seeds from wild species are sometimes conserved, although less commonly, as they are difficult to regenerate. In addition, bananas and their crop wild relatives are conserved in situ, in the wild natural habitats where they evolved and continue to do so. Diversity is also conserved in farmers' fields, where continuous cultivation, adaptation, and improvement of cultivars is often carried out by small-scale farmers growing traditional local cultivars. Nutrition. Bananas. Raw daily value, nutritional value per 100 g, 3.5 ounces, energy 371 kilojoules, 89 kcal, carbohydrates 22, 84 g, sugars 12, 23 g, dietary fiber 2, 6 g, fat 0, 33 g, protein 109 g, vitamins quantity thiamine, B1 0, 0, 31 mg, riboflavin B2 0, 0, 73 mg, niacin, B3 0, 665 mg, pantothenic acid, B mg, vitamin B 6367 mg, folate, B9 20 mu, choline 9, 8 mg, vitamin C 87 mg, minerals quantity iron 0, 26 mg, magnesium 27 mg, manganese 0, 27 mg, phosphorus 22 mg, potassium mg, sodium 1 mg, zinc 0, 15 mg other constituents quantity water 7491 g. Full link to USDA database entry values are for edible. Portion percentages estimated using US recommendations for adults, except for potassium, which is estimated based on expert recommendation from the national academies. A raw banana, not including the peel, is 75% water, 23% carbohydrates, 1% protein, and contains negligible fat. A reference amount of 100 grams, 3.5 ounces, supplies 89 calories, 24% of the daily value of vitamin B6, and moderate amounts of vitamin C, manganese, potassium, and dietary fiber, with no other micronutrients in significant content table. Although bananas are commonly thought to contain exceptional potassium content, their actual potassium content is not high per typical food serving, having only 12% of the daily value for potassium table. The potassium content ranking for bananas among fruits, vegetables, legumes, and many other foods is medium. Uses culinary fruitsy also. Cooking plantain and list of banana dishes. Bananas are a staple starch for many tropical populations. Depending upon cultivar and ripeness, the flesh can vary in taste from starchy to sweet and texture from firm to mushy. Both the skin and inner part can be eaten raw or cooked. The primary component of the aroma of fresh bananas is isoamyl acetate, also known as banana oil, which, along with several other compounds such as butyl acetate and isobutyl acetate, is a significant contributor to banana flavor. Plantains are eaten cooked, often as fritters. Pisang goreng, bananas fried with batter, is a popular street food in Southeast Asia. Bananas feature in Philippine cuisine, with desserts like maruya banana fritters. Bananas can be made into fruit preserves. Banana chips are a snack produced from sliced and fried bananas, such as in Kerala. Dried bananas are ground to make banana flour. In Africa, matoke bananas are cooked in a sauce with meat and vegetables such as peanuts or beans to make the breakfast dish katogo. In Western countries, bananas are used to make desserts such as banana bread. Getty images banana curry with lemon, Andhra Pradesh, India pisang goreng fried banana in batter, a popular snack in Indonesia. Banana in sweet gravy, known as penget pisang in Malaysia. Banana flowers, also called banana hearts or banana blossoms, are used as a vegetable in South Asian and Southeast Asian cuisine. The flavor resembles that of artichoke. As with artichokes, both the fleshy part of the bracts and the heart are edible. Banana flowers and leaves on sale in Thailand. Kilawin na puso ng saging, a Filipino dish of banana flowers. Leaf main article. Banana leaves are large, flexible, and waterproof. While generally too tough to actually be eaten, they are often used as ecologically friendly disposable food containers or as plates in South Asia and several Southeast Asian countries. In Indonesian cuisine, banana leaf is employed in cooking methods like peps and batak. Banana leaf packages containing food ingredients and spices are cooked in steam or in boiled water, or are grilled on charcoal. Certain types of tamales are wrapped in banana leaves instead of corn husks. When used for steaming or grilling, the banana leaves protect the food ingredients from burning and add a subtle sweet flavor. In South India, it is customary to serve traditional food on a banana leaf. In Tamil Nadu, India, dried banana leaves are used as to pack food and to make cups to hold liquid food items. Banana leaf as disposable plate for chicken satay in Java, Nicaraguan nakatamales. In banana leaves, ready to be steamed trunk main article. Banana pith, the tender core of the banana plant's trunk, is also used in South Asian and Southeast Asian cuisine. Examples include the Burmese dish mohinga and the Filipino dishes in Ubaran and Katios, manok, kagubed. King Uak, a northern Thai curry of the core of the banana plant paper and textiles. Further information. Manila hemp, manila paper, and banana paper fiber harvested from the pseudostems and leaves of the abaca banana, Musa textilis, and other bananas have been used for textiles in the Philippines since ancient times. Archaeological evidence of cloth-weaving tools like spindle whorls date back to the period between 1000 BC and 500 AD in the Philippines. However, the tropical environment and the sparsity of pre-colonial records makes it hard to trace its antiquity. Nevertheless, Abaca bananas are the main source of fibers for traditional textiles, still woven among various ethnic groups of the Philippines. Examples of abaca-based textiles include the tinalak, made by the Tibali tribe of South Cotabato, and dagme, made by the Bagobo people. Traditional abaca cloth collected from the Philippines during the Spanish colonial period is found in museum collections around the world, like the Boston Museum of Fine Arts and the Textile Museum of Canada. The Banton burial cloth, c. 1200 to 1300 AD, of the Philippines, the oldest surviving example of banana textile, and the oldest example of warp ikat weaving in Southeast Asia. It is made from abaca, a species of banana endemic to the Philippines. The oldest surviving example of textile made from banana fibers is the Banton burial cloth recovered from a coffin in the sacred Ipat cave of Banton, Romblon, Philippines, and dated to around the 13th and 14th centuries. Abaca textiles were also mentioned numerous times in Spanish colonial records in the Philippines since the 16th century, eventually acquiring the Philippine Spanish name Medrinac, entering the contemporary English language as Medrinax, Medrianax, Medrianacus, and Medrinicals, among other names, and the English name Manila hemp. Aside from indigenous clothing for native Filipinos, Medrinac was also used during the colonial era as canvas for sails and for stiffening clothing like skirts, collars, and doublets. The inner fibers are also used in the making of hats, including the Manila hats, hammocks, matting, cordage, ropes, coarse twines, and Manila paper. By the 19th century, abaca fiber had become one of the most important economic exports of the Philippines. They were in demand due to their strength and saltwater resistance. Outside the Philippines, abaca was first cultivated on a large scale in Sumatra in 1925 under the Dutch, who had observed its cultivation in the Philippines for cordage since the 19th century, followed up by plantings in Central America in 1929, sponsored by the U.S. Department of Agriculture. It also was transplanted into India and Guam. A similar tradition of weaving banana textiles from Musa Basju and Musa balbisiana also existed among the non-Han minority groups in southern China since at least the Han Dynasty, 202 BC, 220 BC. Both fruit-bearing and fibrous banana species have been used. This textile tradition, along with the banana species Musa Basju, Japanese basho, Chinese letter, was introduced to the Ryukyu Islands of Japan at around the 14th century. In the Japanese system of Kajoka Bashifu, leaves and shoots are cut from the plant periodically to ensure softness. Harvested shoots are first boiled in lye to prepare fibers for yarn making. These banana shoots produce fibers of varying degrees of softness, yielding yarns and textiles with differing qualities for specific uses. For example, the outermost fibers of the shoots are the coarsest and are suitable for tablecloths, while the softest innermost fibers are desirable for kimono and kamishimo. This traditional Japanese cloth-making process requires many steps, all performed by hand. Banana paper can be made either from the bark of the banana plant, mainly for artistic purposes, or from the fibers of the stem and non-usable fruits. The paper may be handmade or industrially processed. Tinalak cloth of the Tiboli Dreamweavers, one of the many types of traditional abaca cloths in the Philippines. Abaca fibers being stripped using traditional methods in the Philippines, weaving looms, processing Manila hemp fabrique. Modern Manila hemp bagather uses the large leaves of bananas are locally used as umbrellas. Banana peel may have capability to extract heavy metal contamination from river water, similar to other purification materials. Waste bananas can be used to feed livestock. As with all living things, potassium-containing bananas emit radioactivity at low levels occurring naturally from the potassium-40 K40 isotope. The banana equivalent dose of radiation was developed in 1995 as a simple teaching tool to educate the public about the natural, small amount of K40 radiation occurring in everyone and in common foods. Potential allergic reaction individuals with a latex allergy may experience a reaction to handling or eating bananas. Cultural roles bananas used in puja in the Hindu festival of Ch Hath in northern India. Arts. The Edo period poet Matsuo Basho is named after the Japanese word Chinese letter basho for the Japanese banana. The basho planted in his garden by a grateful student became a source of inspiration to his poetry, as well as a symbol of his life and home. The song, Yes, We Have No Bananas was written by Frank Silver and Irving Cohn and originally released in 1923. For many decades, it was the best-selling sheet music in history. Since then, the song has been re-recorded several times and has been particularly popular during banana shortages. A person slipping on a banana peel has been a staple of physical comedy for generations. An American comedy recording from 1910 features a popular character of the time, Uncle Josh, claiming to describe his own such incident. The banana's suggestively phallic shape has been exploited in artworks from Giorgio de Chirico's 1913, painting the uncertainty of the poet onwards. In 2019, an exhibition of Natalia LL's video and set of photographs showing a woman sucking on a banana at the National Museum in Warsaw was taken down and the museum's director reprimanded. The cover artwork for the 1967 debut album of the Velvet Underground features a banana made by Andy Warhol. On the original vinyl LP version, the design allowed the listener to peel this banana to find a pink, peeled banana on the inside. In 1989, the feminist Guerrilla Girls made a screen print with two bananas, intentionally reminiscent of Warhol's, arranged to form a zero to answer the question in the artwork. How many works by women artists were in the Andy Warhol and Tremaine auctions at Sotheby's? Italian artist Maurizio Catalan created a 2019 concept art piece titled Comedian, involving taping a banana to a wall using silver duct tape. The piece was exhibited briefly at the Art Basel in Miami before being removed from the exhibition and eaten without permission in another artistic stunt titled Hungry Artist by New York artist David Datana. Religion and folklore. Nangtani, the female ghost of Thai folklore that haunts banana plants in India. Bananas serve a prominent part in many festivals and occasions of Hindus. In South Indian weddings, particularly Tamil weddings, banana trees are tied in pairs to form an arch as a blessing to the couple for a long-lasting, useful life. In Thailand, it is believed that a certain type of banana plant may be inhabited by a spirit, Nangtani, a type of ghost related to trees and similar plants that manifests itself as a young woman. People often tie a length of colored satin cloth around the pseudostem of the banana plants. In Malay folklore, the ghost known as Pontianak is associated with banana plants, Pakak Pisang, and its spirit is said to reside in them during the day. Racial signifier. See also List of ethnic slurs section. Banana and racism in sport. In European, British, and Australian sport, throwing a banana at a member of an opposing team has long been used as a form of racial abuse. The act, which was commonplace in England in the 1980s, is meant to taunt players of Black African ancestry by equating them to apes or monkeys.